few years ago, and believe in biochemistry. I think I heard his thesis presentation of his thesis. And then he went off to Lawrence Berkeley Labs, and that particular laboratory has one of the world's leading cancer research groups, and he spent some time there as a postdoc fellow. And then UNM recruited him to come back and to be in his current job. So I've sent out 2 versions of Kurt's talk. The one he sent me, and then I sent out on Saturday morning the truncated version. So anyway, Kurt, go ahead. And,
all right.
All right.
Well, thank you so much. Yeah, so let's see. I want to head and share my screen. So I'll need to just activate this screen sharing component. And then we'll go ahead and pop the PowerPoint up here. So let's see. Yeah, I still can't share yet.
All right, now.
There we go. All right, it just popped up. Okay, so let's go ahead and I'll activate this. And I'll just minimize so I can see everybody. Okay, so I can't see anybody and I can't see the chat. So if anybody, I'll let John, you can kind of monitor the chat for me. And
be glad to.
Yeah, if you see any questions pop up, and anybody, just really feel free to unmute yourself. And I mean, we have a kind of a small group here, so it's actually very nice. So we can have more of a conversation, and I can walk you through some of the things we do. I really appreciate the opportunity to kind of share our research with you today. And as you mentioned, yeah, I had the benefit of, when I came back to UNM, it's kind of atypical. I applied for one faculty position and UNM was it. We really wanted to come back to New Mexico, and so we could be next to our my parents, and we just loved Albuquerque. So that's what brought us back. And we were sick and tired of paying those Bay Area prices. So let's see. So here we go. Yeah. So what we're looking at here in the background, these are all images of normal breast tissue. And I'm going to talk today just about when I first entered the field and understanding the different cell types that compose the tissue and everything that we did to really tease apart each single cell type. And that's why the title says cell by cell, because we were at the time, for the last 30 plus years, the focus has really been on like either the stromal cells or the epithelial cells. And the techniques that were used to really isolate those cell types really focused on maybe 3 or 4 different cell types. So first, before we get into all that, I'll just, just kind of start off very basic. What does the tissue look like? What is it composed of? This is a picture, like I said, of normal breast tissue. This is from a reduction mammoplasty tissue. So in the breast cancer field, we have the benefit of women having surgeries to remove parts of the tissue. And so we have a very nice baseline of having access to normal tissue for study, which is untrue. Like, not many people want to give up part of their liver or pancreas or prostate, that kind of thing. So we benefit from having access, regular access to normal tissues, which then go and serve as models for these other tissue systems. So what we have here is normal breast tissue, and it's colored. It's kind of colored like we use antibodies, and those antibodies go and bind specific proteins, and we can color them with different fluorescent compounds. And so this is just a 3-color image. And the blue is actually a dye. It's called DAPI, and you just put the DAPI on the tissue, and it goes and it really has an affinity for the nuclei of the cells. And so that's the center that holds all the DNA. And it also gives you a sense of scale on when you're looking at an image. You can kind of tell the size and how far zoomed in and out you are of a tissue based on the size of the nuclei. And you can, I believe you can see my cursor here. So you can see there's like one cell, there's another one here and another one there. And the green ones, those are colored with keratin 18, and the red is a panlaminin, just different proteins that identify different parts of the tissue. And this area right here, this is the business end of the tissue. It's a breast lobule. It's also called a terminal ductal lobular unit. And it's where milk is produced in a lactating woman. This is a non-lactating tissues from a non-lactating female. And so, but the milk would actually be produced in all these different areas, in which case it would go down this collecting duct and into a larger duct. That's kind of what I call the milk superhighway, right? So it's a lactiferous duct. And there's a couple other things you can tell from this tissue. This is a three-dimensional image. So it's not typical to get something that looks like it has so much depth to it. This is a very thick image that we had to process over several days in order to get that depth. But what it does is you can kind of see the three-dimensional aspect to it. So you can see that it's kind of invaginated right here. It's kind of like a straw or a tube that you kind of crimp together, or sometimes like a penne noodle, you know, it's kind of squished and it's fluted. And so the reason for that is when milk is being made, that allows for the expansion of the milk. And so the ducts are just designed to be able to expand and hold the milk and allow the milk to go towards the nipple. And these cells, these red ones, are also have smooth muscle actin on them, and they'll contract, and that'll help push the milk towards the nipple. All right, so you also think in a three-dimensional aspect. So if we took a little cube here and did a cross section of it, this is what it would look like. And typically you would see these things on a very thin section where you take the tissue and do very thin sections and you can stain it. So this is what it would look like, just little tubes. And that's all very normal looking tissue. Now, one of the main things, there's a lot of questions that you get is what is, when we're talking about breast cancer, the differences between invasive cancer and DCIS. You'll hear ductal carcinoma in situ. So if you have abnormal growth of these cells, they will fill the lumen. The lumen's the center tube area of these ducts. And that abnormal cell growth will fill in. And if it's the pathologist, if you had developed a lump or this was detected on a mammography, typically would do a biopsy and determine if there's any areas where these cells have left. But if they're all contained within the duct, then that's considered in situ, in place. And that actually is a good sign, because DCIS with treatment, the prognosis is really very good. And even to the point of where there's cancer when in UCSF, surgeons, a very prominent surgeon in a panel that really would like to change get the carcinoma out of that name, because it's just so scary when you get that diagnosis, yet 99% of people do very, very well. So now the bad thing that happens is sometimes this could fill up, and if those cells cross this barrier, which is a basement membrane barrier, those cells will grow, continue to grow. They can invade into what's called the stroma of the tissue, and eventually they could enter blood vessels and metastasize and that kind of thing. So this would be called invasive ductal carcinoma, or invasive breast cancer. Most common one is invasive ductal carcinoma. And that the prognosis depends on all kinds of different factors, like the size of the tumor, whether or not there was any lymph nodes that were detected. But it also doesn't occur overnight. This is typically believed it could, years and years for these things to occur. So that's the other thing that the surgeon in UCSF told us, would say, breast cancer, it's not an overnight emergency. So it's always good to go get second opinions and seek counsel of others, especially in the cases of DCIS. Okay, so that's all nice cartoons, and I really had fun with my pen, so I was going to town using that. So this is what's called an H&E section. It's hematoxylin and eosin. Hematoxylin is the blue stain, eosin is the red, and these are dyes are used to stain tissue so we can actually see them. Otherwise, they just look clear, but they bring out different aspects of the tissue. So this is a low power image of normal breast tissue, and we can use our microscope, switch objectives, and zoom in on certain areas. So we can do that again, and we can do it again. And now we actually start to be able to make out some of these nuclei. So these are the nucleus of the cells, right, that are blue. And we can see that these cells right here are the luminal epithelial cells because they form the lumen of the duct. And it's actually kind of difficult without having the antibody stains, certain proteins, of determining which one's a luminal cell and which one's a myoepithelial cell. But generally, you could say, all right, all these internal ones that we're just going to color green here, those are going to be the luminal epithelial cells. And then the myoepithelial cells are on the outside. And of course, they're called myoepithelial cells because myo, muscle, they do have smooth muscle. That's one of the key markers of this cell type because they also are responsible for contracting and expelling milk. And then another major cell type that is commonly recognized are the fibroblasts. And so this is collagen right here, this pink area. And so these fibroblasts are responsible for making collagen, and they're out in what's the stroma. And so there's also a basement membrane that surrounds. And so these epithelial cells sit on a basement membrane. And that's really, when you have a DCIS or invasive cancer, it's really the integrity of that basement membrane is important. And just for comparison, this is normal on the right here. And then this is an invasive carcinoma. And clearly you can see a huge difference between this organized, beautiful tree-like structure and this just disorganized growth of cells. And that's commonly what we all refer to as tumor, is just uncontrolled cell growth. Okay, so my laboratory, we focus on a fundamental question in biology, and that's how do cells form and maintain tissues? Really, when I wrote this, and I had a student right there, is like, well, really, I don't mean cells. What I mean are how do the different cell types that are in the tissue coordinate to form and maintain tissues? That's a huge, encompassing, enveloping problem and question that we're kind of dealing with. And also, it's not just the normal context, but also how do these different cell types contribute to the malignant process? And so over the last 20 or 30 years, we've grown in our knowledge of tumors and understanding how they're formed and the genes and oncogenes that cause uncontrolled cell death or uncontrolled cell growth, but also that there's other cell types in the what's called the microenvironment. There's fibroblasts and blood vessel cells and different kinds of epithelial cell types. And those all can either apply the brakes or gas and fuel tumor growth. And that's not really that well understood. So back in the early 2000s, one of my big questions, and it wasn't like a big paper or textbook you go to say that would really lay out all the different cell types that are in the tissue. And so what I did is I got all kinds of different antibodies, and we got normal tissue and sectioned them and applied them to the tissue to see essentially what we could see and determine how we could come up with a method that would identify the different cell types in the tissue and how we could apply another technology that would allow us to break those cells apart and actually sort them into different groups so we could go and do further analyses and experiments on them. And so this is just where we can kind of zoom in again on a tissue where we stained. And we can start to make out, as we get closer and closer, different cell types. And I think I have one more zoom in right here. And so again, this is a duct, and it's got to see these luminal cells right here. Smooth muscle actin, again, is labeling these myoepithelial cells. And also what we have is panlamina. Now that's the basement membrane I was talking about. So you can see this intact basement membrane that these myoepithelial cells sit on. But it also, the panlamina is labeling this thing right here, and that's a capillary. And so if we back up, you can see that there's capillaries, and this is a capillary right here, here. And so you can see a tight association with the epithelial structures. And I'll show you another picture in just a moment. So we can use these different, so what eventually developed and got nice images, and we can use these antibodies to identify different cell types. So we'll just walk through the 12 different cell types that we identified. One of them was the luminal epithelial cells. These were well known, so it's not like we discovered these, and the myoepithelial cells. Okay, we did discover a rare epithelial cell type that was really interesting. It was kind of fell outside of the epithelial compartment. We're not still quite sure what it does, but we called that one population 4 because it was the 4th one we identified. We got really creative with these names, but it does help because when we identify these cell types, it's usually identified with 9 different antibodies in this other flow cytometry experiment. And so it got really old saying, CD34, putting all those markers down to where we just said, okay, this one's number 1, this one's number 2, this one's number 3. And so we called this one number 4. And number 5 is vascular smooth muscle cells. And these are the cells that surround large vessels. And we kind of lucked into identifying this one. Now, it's not like we identified vascular smooth muscle cells, but what we did do is identify a way of sorting the cells into a pure population. And that actually is the same thing with pericytes. Pericytes are notoriously heterogeneous, and it's difficult to identify them in different tissues. And the panel that we used we're able to get pure pericyte populations, and that's actually quite a contribution. And in this image too, now we're zoomed in, and you can tell again we're zoomed in because the nuclei are really big. And this green area right here, that's a capillary. And so the capillary is made by vascular endothelial cells. And then the pericytes, they're perivascular cells. So these are cells that sit on capillaries and they wrap around them and they kind of maintain structure. And they're also believed to help the communication between the different endothelial cells that they're surrounding. They're also really well known in the brain because the pericytes are what are responsible for forming the blood-brain barrier, because you get a tight pericyte coverage of the vessels. Another cell type, number 7, all right, this is going to be the lymphatic endothelial cells right here. So you can tell the difference between lymphatic endothelial cells and these right here, these red ones, that would be a regular endothelial and capillary right here. And these are lymphatic vessels. And as we know, the lymphatic vessels, those are the vessels that kind of collect fluid from the interstitial spaces, these spaces in here, and they go to lymph nodes. And they collect things in the environment. So they're kind of the vacuum cleaners of the tissue. And the things go in, and then they can't get out, and then they go to a lymph node. And in the lymph node, antigens and things are presented to the white blood cells that then elicit immune responses. Okay, so then that's why you look at lymph nodes when you have a tumor. We want to know if there are any cancer cells that may have gotten into the lymphatic vessels. And then the very first pit stop would be a lymph node. And so that's why you look at lymph nodes. Adipocytes. So this is also a nice image of the adipocytes. They're kind of, you can see the little nuclei right here on the surface. They're just filled with triglycerides and fat. And so that's why they look so big. And then let's see, well, fibroblasts. So we have fibroblasts right here. You can see that they're just all over the place. And then we also have white blood cells. So these are leukocytes. And surprisingly, we found white blood cells. They were all throughout the tissue. They were usually associated with the epithelial cells. They were in the stroma. And so these would be macrophages. They're kind of sitting there waiting for pathogens to come in. Remember, we kind of have to think of the breast as in here would be connected to the outside world, because at the nipple, you have an entrance into the body. And so this area right here has a huge immune surveillance. So you have a lot of white blood cells that are going through the epithelial spaces, and you'll often find white blood cells in the lumen of the ducts. And then for those of you keeping count here, we also identified or were able to separate out 2 different kinds of luminal epithelial cells. The first one are estrogen receptor positive, and then estrogen receptor negative. This is also well known. It's not like we discovered it, but we called the estrogen receptor number 1 and estrogen receptor number 2 populations. And that really rounds out the 12. And if you are keeping count, red blood cells, those were our number 12 or number 11, which I don't have an image of. And so if we just kind of take a step back from the tissue, this is a very low power scan image of breast tissue. Gives you a very nice picture of the landscape. And we can see these epithelial areas here and here. Here, they're just scattered all throughout. And then what I've done, this is in our paper, but what we've done is we've labeled the cell types that you would expect in these areas. So 1, 2, 3, and 4, these are all epithelial cells. You'd expect to see pericytes and endothelial cells and fibroblasts, and you'd expect to, in those areas of capillaries, expect to find white blood cells and red blood cells. Then we have vascular smooth muscle cells. It's really hard to see. It's probably something under here, some capillaries, lymphatic vessels. What's really clear here are adipocytes in this area. You can see a very large collecting duct. This is a lactiferous duct. And so all these different smaller ducts will connect up to a main larger duct that eventually goes, and they kind of branch to the nipple. And what's really impressive too is see all these capillaries and how they encase the epithelial area. So if we zoom in on a different tissue and just look at it, they just surround the epithelial tissue. So they are centered there. And then we can kind of take a step back and say, well, okay, that's all nice, well and nice, and you've shown us nice pictures of all these different cell types. But what we're also doing at the same time is coupling it with another technology to where we could isolate, purify, and culture each one of these different cell types. And so that is called flow cytometry and cell sorting. So I'm not sure if this is not, I mean, it's a common thing in the biomedical field to do this. So I figured I would kind of show you what this does. And so if we have the breast, women have reduction mammoplasty tissue. And so what we get and acquire are just kind of a chunk of tissue. And then what we do is we use collagenase and enzymatic digestion, and that will give us these single cell preparations that are just in a tube where the cells are floating. And what we do then is we add a cocktail of our antibodies to that. And these antibodies are labeled with different fluorescent compounds. And they will label the cells different colors. And then what we can do is then take it over to what's called the FACS machine. It's the fluorescence assisted cell sorting is what that stands for. So it's a sorting machine. And so how does a sorting machine work? Well, if we start with our colored, this is where the physicists here and people, this is kind of cool. A lot of this was actually developed at Los Alamos National Laboratory, the flow sorting technology. And so the cells here, they'll be colored with different fluorescent compounds. In the machine, it mixes it with a saline solution, and it's a very stable laminar flow that it goes through a nozzle up here. And what happens, these are supposed to be my little lasers that I have. So it's going across the stream. And if the cell with the laminar flow is in the middle of that stream, it'll hit the laser. And then you have detectors that are 90 degrees to this in the machine, and it goes through a set of optics. And so you can break this down. Now the cytometers these days are capable of detecting upwards of 25 to 30 different fluorescent compounds. The one that I used could detect, it had a limit of 8, and the one I currently use at UNM has the ability, I typically use about 12, but it has the ability to detect 16 different fluorescent compounds. So got a lot of capabilities. Our antibody panel is like 9 different antibodies. And so what happens is a cell will hit, and if it has that antibody on it, it will fluoresce in that color. The machine will detect it. And down here, the machine is keeping track of this constant speed, and it will say, hey, this cell right here is the one you're interested in. And it passes through these electromagnetic plates. It will apply a charge through a little kind of pin up here in the stream that will then charge the stream, and then the plates will, the one that I use can sort into 4 different tubes. And so we can sort groups of cells and then do another sort and get pure population. So at the very end, we've sorted off all the blue ones, red ones, orange ones into a pure population. And so that is just amazing to watch because it does this at about 10,000 cells per second. So when I have my students, I'm like, how would you like to do that by hand? Like, oh, 1, 2, it's amazing. It's a nice piece of technology. And so, as I said, our goal was to develop a comprehensive sorting strategy where we could detect every single cell type in the tissue, isolate them and sort them and use them for experiments. And so at the end of what took 4 or 5 years, we were able to do that. And so this is the sorting all the different 12 populations that we get. Just to kind of show you a little piece of data, we'll kind of lift up the hood here for a moment. Don't worry about all these markers and things. We're just going to focus on one of them just to kind of give you a little bit of example. But we do go through different steps. And what we're doing is we're drawing what's called a gate, essentially an electronic box in the software, and saying, hey, I'm interested in this group of cells right here. And then go ahead and take these cells and I want to analyze them on another plot. So that's how you get the multi-dimensional aspect of all those antibodies, because you can look at them in different dimensions and say, all right, I'm interested in these CD34 positive. So these are, each dot here is a cell that has gone through that laser beam. And in this case, we're looking at this marker that's called CD49F. And then another marker over here, it's CD34. And so we can see these cells right here have both CD49F and CD34. These have CD34, but no CD49F. And then all these down here are negative for CD34. And so this very first step is I take these CD34 positive cells, we look on another graph, and you can see how we break them into different populations. These are actually pure at this point. Any questions? Pause for a moment. This I usually, even the scientists are like, wait, what? Hold on. So don't feel bad. But the whole point of this is we were able to, just want to show you actual real data that, let's see here. Okay, kill that. Here we go. Okay, so we're able to sort the cells into pure populations, show you some real data in doing it. So we'll kind of put the hood back down, looking at these different cell types. So our ability to sort cells into different batches now allow us to attempt to grow them in the dish. And so I came from, like John said, Lawrence Berkeley National Laboratory. That's where primary cell culture was developed in the late 70s, early 1980s by a woman, Martha Stamfer, who just amazingly be able to grow these cell types. And really, since that time, two major cell types are used in the field from normal tissues. They were essentially myoepithelial cells. They're also called human mammary epithelial cells, where they wouldn't sort, but they would put the cells into, or the tissue chunks into a dish. And really, the myoepithelial cells grow so fast, they pretty much overtake the culture after several what are called passages, where you kind of split the cells. Another one were fibroblasts. And so you could use different techniques of, say, differential trypsinization and things that would kind of get a pure population of fibroblasts growing. Now, of course, there's laboratories throughout the world that kind of specialize in different cell types, and they come up with different ways of sorting and enriching or purifying for their cultures. But by and large, 2 or 3 different cell types are the main ones that have been used. And so we're able to use flow cytometry and contribute all 9. We grow these now 9 routinely. Now, the ones that have never been actually published before were the pericyte, like a pure population of the pericytes from breast tissue, and lymphatic endothelial cells from breast tissue. So those are completely novel that we did develop. So how can we be certain of these cell types identities? That's right. So during the development, a lot of times we get the cells growing, but we're always worried because some cells grow faster than others, whether or not it's just another cell that is contaminated and that we're growing. And so what we would often do is stain the tissues with markers of the different cell types. And then that would give you the confidence that tell you that, yeah, no, you have a very pure population of a distinct cell type. Another way is to use omics technologies, and that's Rna expression. And so, for example, I've shown you that we can take breast tissue, isolate the tissue into different cell types, and create these primary cell cultures. So what we've done throughout the years is we can take those pure cells and extract their RNA. So we've heard a lot about RNA over the last couple of years because of coronavirus, where RNA, remember, is the copy of the DNA. So every cell has the DNA, that's the human genome. And then you can think of that as the library, right? So, and then each kind of cell will take from that library and read different things. And that's the mRNA. And those are the instructions to code a protein. And so by looking at the different kinds and amounts of RNAs that a cell is making, that gives you some insight into what that cell is doing. And so what we do often is we use a kind of a very broad technology called RNA sequencing, where we can look at every single RNA that is within a cell. We can also do it. So we've done that on freshly sorted, uncultured cells. We've got primary cultures where we've looked at RNA sequencing. And we can also look at the proteins also. That's called a mass spectrometry, where we can look at the proteins and make comparisons between the RNAs and the proteins. And there's a new technology called single cell RNA sequencing where you can take a batch. So what we've done is taken a batch of pure cells of, say, a population of pericytes, and you can look at the RNAs from each individual cell that's in that population. And so this is very useful looking at heterogeneity within a population. And so you can see little things coming out here. So there is some heterogeneity, and this is a pericyte population here. They're all quite expensive too. So this one is about $20,000 to do the deep RNA sequencing. The primary cultures is another like $15,000. The mass spec was about $15,000, and the single cell is about $7,000. So it's a lot of money to do these kinds of, but it's so informative, and we are just up to our ears with data. And so really, I like to kind of show just some examples of the way that you can use this. One of them is you just let the computer, you go into the program and you say, here's the RNAs, and because differentially expressed, these are genes that are different between cell types. And if we're trying to understand differences between cell types, it's common to say, all right, I want the ones that are really different between cell types. And then just show me the different populations that are sharing those gene expression. And it'll just, what's called an unsupervised hierarchical clustering. And it just groups cells based on a whole bunch of different genes. And what it did is very nicely recapitulates how we sorted them. And so each bar here up at the very top, this is an individual sample. So 10, for example, is a white blood cell population from one patient, and then that second line is a second patient and a third patient. And so the clustering actually said, all right, for example, all these red ones, they are all kind of very similar. They're kind of close to these green ones. And if you look at the blues, and like, all right, these luminal cells, they're kind of similar to these. So that's very nice. So yeah, that's actually what we would anticipate. And so that is just a way of validating your data set and saying, yes, we are getting what we think. The machine is identifying genes that are differentially expressed and putting groups of cells into populations that we can make sense out of. And for example, we can pick out little populations. So this is a heat map where we're looking at very low expressing genes. And then if it's expressed at a high level, it's red. So we can see this little patch of red that is under 7 and 9. If we look at 7 and 9, these are lymphatic endothelial cells and vascular endothelial cells. And there's a bunch of genes in here. They're not all being shown. But if we just pick out one of them, here, selectin P, which is known to be expressed in endothelial cells, you can now look at this graph right here. This is the actual mRNA level. This is on a log scale with log 2 values right here. And then this is a linear scale over here. So you can see that, yes, indeed, this gene is being made by endothelial cells. And so you can look at other genes that kind of share a similar pattern that are within this box right there. And for example, we can show another one, like here's keratin 15, identifying luminal epithelial cells. So this just, yeah, I guess I got one more, white blood cells. So this is actually a marker that is on T cells, CD8. And so, yes, we get a little patch of specific expression within T cells. All right, so that's nice. We get validation, but what can they really tell us about each cell type? Because you really don't want to go through one by one of all the, I have gone through, but it is very time consuming. And so are there other technologies that we can use to really get some sense of what the cells are doing. And this is where my laboratory splits into two different things, where we explore normal biology, where we're interested in what genes and processes define each cell type, and actually communicating with each other and what are the consequences of different cell type interactions. And then also on the tumor side, obviously, our whole idea of really wanting to understand the normal biology of these cells is so we can understand how tumors grow and what regulates the growth of tumors, and why some tumors seem to just sit and not really do much, and others will grow and metastasize and cause problems. And so the bread and butter, which is the basis of my grants and trying to get funding for the laboratory, really trying to understand, now that we have these novel cell models, of which of these cell types have any kind of function or do anything to the tumor cells and affect tumor progression. Okay, so starting with on the normal side, we can take all that gene data, and what we do is we look for differences, and it's very similar to the hierarchical clustering, but what it's now doing is we're compressing everything down onto a two-dimensional space. And so it's just a linear model in algebra, like the linear. So what we're doing is we're drawing a vector through the data set and looking for the genes that explain the most amount of variance. And so, for example, principal component 1, which is on this x-axis, so it kind of goes this way and this way. So we can say things on the left side are epithelial and really high in the luminal epithelial, that's the blue. And then on the right side, these are going to be the things that are mesenchymal. So we can see these fibroblasts, pericytes, vascular smooth muscle cells, etc. And so, yeah, if we look at the gene and in that principal component, we can look at what genes are contributing to that, and we can look at all these genes and see, oh yeah, all these blue ones have really high expression. So it did a really good job of pulling them out. And I've put stars on the ones that we recognize and use as marker of luminal epithelial cells. So it's doing a very good job of pulling these out. And then we can look and say the second, what's called the second principal component. So this first one explains 33% of the data between all cell types. And this one explains 17%. So each component explains fewer and fewer variants. So doesn't really matter. All we are doing is we're looking at the genes in that principal component that are either at the top or bottom of that list, because those are the ones that are contributing most to them being sorted out. And those are going to be the genes that are doing distinct things. And we can look at those genes and say, all right, well, what are these genes doing? And that's actually what we do. So what we do is we take, say, the top couple hundred of those genes, and we apply them into a filter, a database. And what it will do is say, you know, these genes, and it does the statistics on it, and say, well, you supplied me with, say, 200 genes, and statistically speaking, based on you supplying a gene set of 200 genes, you had a really high significant hit that a lot of the genes that you supplied are involved in, like number 1 here, cell adhesion, molecular binding, cadherin binding. And you just go down these lists. And so that's where the rubber hits the road, where now you get these ideas of, okay, the genes, it seems like keratinization is important. That's not surprising. But other ones, say like peptidase activity, that's where you have to start doing experiments and determining, well, what is it doing? Why are these cells making these proteins? And if you look at these genes that were identified, you can see, yeah, sure enough, they're being made by these blue cells. So you can see that the blue is always at the top of this list here. So these are processes that seem to be enriched in these different cell types. Okay. Another way of doing this is, because we have RNA sequencing, we have a list of every single gene in the genome, and we know in every single cell type the level that gene is being made. And the way that this technique works is you put everything in when you compare 2 different cell types. So in this case, we're comparing the 2 different luminal epithelial cells. So one of them is ER positive, that's this light blue over here, and ER negative. And what this is supposed to represent is every single gene, like 20,000 genes, going from left to right, where the genes that are highest in ER positive are on the left, and the genes that are highest in the ER negative are on the right. And then what you do is you then go through sets of what are called gene sets. And so we know that in a certain gene set, the gene set I'm showing here is estrogen dependent gene expression. We know the genes that are responsible for carrying out the signals like estrogen receptor and downstream signals. And we know that the genes that respond to estrogen. And so what we're doing is we're saying, in that set, where do those genes fall? Do they fall more on the right, or do they fall more on the left? And you can see that we get a very enriched set of lines. And so if it falls in that set, it puts a line here, and that does this kind of cell counting. doesn't really matter. All it's really doing is saying, hey, you've got a lot of genes that are involved in estrogen-dependent gene expression at the P or false discovery rate of very low. It looks like these cells over here are involved in estrogen gene expression and response. Well, this was a validation experiment. These are ER positive cells, and so that's exactly what we'd expect. And so it's nice to see that this is indeed working as expected. It was able to find, yes, indeed, these cells are responding to estrogen. And you can look at the set and you can say, this is the ER positive cells, the light blue, and then the dark blue are the ER negative, and these are the different genes. And we can pick out genes in this list and say, all right, well, just show me a couple. Like, well, here's the progesterone receptor. Sure enough, the light blue is dominating expression. Here's the estrogen receptor, again, higher in the light blue cells. And if we go down to the lower end of the list, yeah, we don't really see much of a difference. And so, again, it appears to be working very nicely. Okay, so that's just one gene set. Now we've got about 1,000 to look at, and I'm sure you don't want me to go through 1,000 gene sets. So one thing that we do is we say, all right, a lot of these gene sets are sharing genes. So can we use a technology? So this is just clustering where we can say, all right, these different gene sets that were identified are sharing genes. So they seem to be doing similar things or identifying different themes. And so we call these biological themes. For example, here's the one that we just showed, estrogen signaling. So there's actually 3 gene sets that have some kind of involvement in estrogen, and all 3 were actually identified in ER positive, estrogen receptor positive cells. And so this is where we discover things. So you can go through each one of these and say, oh, well, I didn't actually know that, like VEGF signaling, you know, and you have to pick them. And this is where we've got lots and lots of experiments ahead. And, you know, it's going to take a whole community when we release this, get it published for people that are exploring certain areas of these fields of being able to use these cells to explore what the consequences of this gene expression is. But again, for estrogen signaling, we have 2 different luminal epithelial cells. And yeah, so here's the ER positives and ER negative right next to it. And so these have different functions, and we learn by what those functions are by exploring this map. Okay, so with that, that's kind of where we're going to lead off with the normal stuff. But now the tumor biology, like how can we use this data set and these models to explore tumors? And so and we have all these primary cell cultures right here that we can grow in the laboratory, and we do grow in the laboratory and do experiments on. So one experiment that we have is we color tumor cells. So these are breast tumor cells that we colored with something called histone 2 beta GFP. Now that doesn't really matter. The GFP, we colored them with this green fluorescent compound. It's a protein that fluoresces green. The H2B makes it go to the nuclei. So it makes the cells easy to count, essentially. So this is a very common technique used in the field. So we use the flow sorter, and what we do is we deposit 30 cells into what's called a 96-well dish. So it's a cell culture dish that are separated, little wells, so they're separated by each other. And the machine will very precisely put 30 cells into each well. Now on these plates, we have also added either pericytes, or actually every single cell type, fibroblasts, pericytes, so on and so forth. And then also we put tumor cells that don't have anything, okay? And so we image them weekly for like 3 different weeks, and then just take a look at the images. And so, for example, I'm not sure if you can see it, but there's a little patch of cells right here. Those are MCF7. Those are the tumor cells that are marked in green. Now, what you can't see in these areas out here, these are where we added 1,500 different fibroblasts. You can't see them because they're not fluorescent. But the fibroblasts were able to get a little expansion of tumor cell. And that's great, because what I didn't show is the control where you didn't have much growth at all. Now, what was amazing is that in the pericyte populations, we had this huge expansion of tumor cell growth. And that was impressive. And typically, this is where I say, now the scientists out there, you're probably saying, how do I know that you didn't cherry pick the data and just show me 2 really good ones? And you'd be right. How can I be sure that this is reproducible? And so I had an undergraduate say, all right, what I need you to do is in each one of these wells, I need you to develop a software program that will, and pipeline that will count every cell. This should be fairly easy. Those are the famous last words of a mentor, right? Should be easy to do. So 6 months later, he came back. He finally, okay, I got it to work. Encountered so many different problems in which we said, all right, well, you encountered all these problems. We should go ahead and publish it. So we have this small little paper down here that we published of, if you have this EVOS platform, you can use this pipeline to count cells. And what it does is you can zoom in on these, and what the software will do is it'll put a circle around every single cell, and then it'll count it. And so if you're in the microscopy field, this is actually a very common technique to use for counting. And so with that, we're able to say, all right, the control cells, we get this kind of growth. With fibroblasts, we get this kind of growth. And oh my goodness, with the pericytes, we see a huge expansion in these tumor cells. And this is one tumor cell type, MDA-MB231. So what if we use a different one over here, MCF7s? We see exactly the same thing. And these are very different tumor cells, which we use them to see, is it only specific to one kind of breast tumor? It seemed like it's more general. Okay. And so this is where we start doing experiments and saying, all right, well, right now in those previous experiments, we were using 1,500 cells per well. What if we go a little bit above and a little bit below that? Can we see a dose response? And indeed we do, right? So if you add more cells, you get more of a response here and here. So things are getting really exciting, saying, all right, we add more pericytes, we get more tumor cell growth. What of the things that are the question is, do those tumor cells, do we need to have the pericytes or the fibroblasts, you know, the cells, these are done in the same well. Do they have to be there? Do they have to be touching each other, or are they secreting something? And so a very common experiment is to grow in a different dish pericytes by themselves, and then you just take the media, the kind of the liquid that's on them, pick it up, and then go and put that on tumor. So that's called conditioned medium. So this is media that's been grown only with pericytes. And when you take pericyte conditioned media and put it on tumor cells, it also causes the tumor cells. So what that tells us that the pericytes are secreting something that are causing the tumor cells to really expand and grow. And so that makes our life a little bit easier, because now we can stop worrying about these kind of network and interdependent interactions and say pericytes are just secreting something, and can we look and determine what that is. And so what we did here is we took that conditioned media, and so we get a modest response in the conditioned media. What if we heat and activate it and it caused it to go down? And that tells us most likely a protein, because proteins are heat and activated. Okay. And then another thing that we can do is we can take that conditioned media and we can filter it through different filters, and what's called by size. So here, when we don't have anything, we get this kind of response. If we get the pericytes grown with the tumor cells, we get this very large response. And if we take the pericyte conditioned media, it seems to go down a little bit, which makes sense because we're actually only adding this once a week. And so, but we still get a statistical response from having nothing. But then when we sort out this media into different sizes, is that if we look at things that are less than 30 kilodaltons, yeah, we got a baseline response. And I don't know if I didn't show the data. We actually have another one where we have greater than 30 kilodaltons, and we get a nice response showing that it's something that is about 30 kilodaltons or larger. And then this is the very last thing. This is kind of where we are right now, is just saying, well, what could it be? So we've filtered through fibroblasts and pericytes and able to identify potential candidates that are above 30 kilodaltons in size, are secreted by pericytes specifically. And that's what the grant that I have out right now is, to look at some of these compound or proteins to really determine what is the mechanism of this response. And we think that it has a very significant to patients, because if you look at patients' tumors, and what we did is we looked at specific pericyte genes and genes specific to every single cell type. And those that were genes that were unique to pericytes were actually 60% of those genes had were correlated with reduced patient survival. And so that's why we think it was so important to study and look at the mechanism pericyte-induced expansion of tumor cells. And so with that, obviously the people who did the work, myself, and this is my postdoc, and we have graduate students in here and undergraduate students, and at UNM, we have colleagues at Lawrence Berkeley National Labs, Mina Bissell, Dana Peer Labs, who helped with the RNA sequencing work. And with that, I appreciate your attention and be happy to answer any questions you have.
Kurt, could I ask you a question?
Yes, sir.
What exactly are parasites?
Yeah, so pericytes are the cells that line the capillaries. So if we go here, I can show you a picture of them. Oh, screen sharing has stopped. The shared window is closed. Let's see. Oh, I killed it. Okay, so let me find you a picture of them. Right here. Okay, so can you see that?
Mm-hmm.
Right here. So this is a capillary. The pericytes they're called peri-endothelial cells. Mural cells is another word for them, Roget cells. They're cells that line and surround blood vessels, and smaller blood vessels like capillaries. And so you can see this nuclei, this little blue cell right here. The red, that's the pericyte, and the endothelial cell is this green cell in the middle. And so they're just cells that surround capillaries.
And so are you saying that the parasites carry more responsibility for secreting something that makes cancers worse?
In the laboratory, that's exactly the hypothesis.
Okay.
I do have some data, I didn't put it on here, where we use some spatial transcriptomic data where we actually see. So typically you would only see like in normal tissue, you only see the pericytes that are next to a blood vessel. And so we've used techniques to stain for pericytes, and in tumors, we see an expansion of these cells in a tumor. And so that it's plausible to think that, yeah, and then you also see the same thing with fibroblasts. So you get a stromal response, and that's kind of the laboratory where I came out of, is looking at the importance of the microenvironment and fueling and repressing tumor cells. And it looks like pericytes are a subtype of this mesenchymal population in the tissue that in a tumor can expand. And we think that the pericytes are really contributing a lot to causing those tumor cells to grow and migrate.
So in a sense, you're saying that the cancer is a function of parasite function.
Yeah, and actually it's not just pericytes, but it's all the different cells. I mean, so tumor cells it's just like a kind of what we talk about as a tumor organ. It's lost sense of certain controls, but they do respond to certain rules. Just like in developmental biology, you have different cell types that can make certain ligands and cause, for example, HER2, right? So the tumors are making HER2, and you can repress these signals. And there's other cell types that make these ligands and growth factors nearby. And if you get an expansion of those that are kind of in this chaotic mix, they're kind of, and it's also really well known in white blood cells with kind of this kind of chaotic circle where the tumor cells will fuel the white blood cell and macrophage growth, and the macrophages will secrete ligands and hormones that cause tumor cells. So it's not that it's the only one, but it seems to be a very significant one. The pericytes are contributing. And the problem with and so this is kind of the novel thing about the torturous process that I took you guys through of identifying all these different cell types, was that it was able to find this, because trying to identify pericytes in different tissues has actually been historically difficult. And so all the real experts in the field have identified for so long problems with markers and the heterogeneity of these populations, and it's difficult to purify these cell types. And we kind of lucked on it. Well, it wasn't luck, because we were kind of going through methodically identifying cell types. But we did kind of stumble upon pericytes in the flow cytometry, because I'd used a marker for a different cell type, and it actually identified pericytes as well. And that's how we've identified them. Said, hey, this is how you sort them out. And then actually spent a couple years trying to get them to grow. And it's just amazing to see the response. It was very dramatic. I remember coming home and showing my parents, it's like, look, let's get the tumor cells, and it causes this kind of expansion, because it's well known that fibroblasts do that. And so that's why we use fibroblasts as a control in all these experiments. to see that we got improved and increased growth over fibroblasts, and what's well studied in the field is kind of what we think is going to be the innovative part of our grant and hopefully get us over that kind of funding hurdle.
So if you take it from where you put it so far, what would the steps be to figuring out what you do to treat or combat this parasite contribution to the growth of cancer cells?
Yeah, so the first steps, because it's so much in its infancy, is really, so what I outlined in my grant is what we need to do is we need to know more about parasites, first of all. And so sort them and actually look at parasites in tumors. So what I haven't shown you, I've shown you a lot of parasites, or what I have is a lot of parasites in normal tissue. But what we, breast tumors, it's a mixed bag because it's you get all kinds of different types of tumors. So we really need to look and determine good markers where we can do it in a little bit faster way of looking at the parasites and exploring those cell types in different kinds of breast tumors. And then also need to look at, because of the heterogeneity of parasites, is there a specific parasite population or subtype? And that kind of goes to this. I didn't show that data, but the single cell RNA, the reason that I did the single cell RNA sequencing to look at heterogeneity of populations is to determine, are there different subfractions of parasites? Indeed, there are. And so that's another question, is do we see an expansion of specific subpopulations of parasites in one kind of tumor versus another kind of tumor? And then obviously, not just the primary tumor, but where we can have animal models where we look at parasites and tumors in metastases, because that's really what's causing, death. But parasites are everywhere. So we think what we're seeing in the normal tissue and in culture has the potential of actually happening in metastatic sites. And so it'll be important to look at that too. And then obviously identifying the mechanism and the ligand of what it is that parasites are making that are causing that cell expansion, because you'd really want, that's the thing, is like, well, that's great, but what we're really trying to do is target and cure cancer, or at least treat cancer. And what is it, something that we can target with a drug? Is it a pathway that we can target and use inhibitors? Can we specifically target that ligand or the receptor or something like that nature?
I don't mean to cut you off, but we are just about at our time limit.
Mm-hmm.
So if you want to kind of give us a little bit of a closing.
Well, that's it. I mean, so this is the product of the national funding. And, we appreciate the money that people donate to the Komen Foundation and the Department of Defense and National Institutes of Health. These are monies well spent. And so it's kind of just the product of the NIH and funding mechanisms that go to help and support science and health science research. So we appreciate everyone's help and support.
So I have a question that came across. What causes the cells to go rogue?
Yeah, so that's actually quite interesting. So I presume we're talking about tumor cells. And
Yes.
so, underlying all that are mutations. But it's not just mutations. You also have, it's a long process typically, especially in the breast, that when you have these mutations, you actually have also a breakdown of communication between the normal, or the epithelial cell, which if it acquires enough mutations in certain pathways, it will respond. And if you have a breakdown also in certain stromal components, or a breakdown in the myoepithelial cells, where you kind of lose that integrity and loss of normal kind of three-dimensional structure and communication, the rules kind of break down. And then that's what really fuels the process, that it's just a kind of a snowball kind of building up and going downhill.
Thanks.
Mm-hmm.
Thank you very much, Kurt.
Yeah, absolutely. I appreciate having an opportunity to talk with you guys today.
Good. So we had about 12 people online, and I don't know how many are in the classroom, but anyway. 8. Good. So you going to sign us off, Nicole?
I will.
Okay. Take
Okay,
care.
thanks again.
Bye-bye.
Nice talking to you.
Thank you so much.
Yeah, about 20 people.
They didn't remind us that the test is